Next one is cytokinesis. First, we have completed the karyokinesis first of the meiosis first. Now the cytokinesis first. So what happened in cytokinesis? There is a division of cytoplasm simply. So two daughter cells are formed, and each daughter cell is with a single nucleus and consisting single set of chromosome, means now these are haploid. So n is equal to 1c and here n is equal to 1c. Haploid condition have one chromosome because we have taken 2n is equal to 2c, means diploid condition. We have taken diploid condition as a 2 chromosome. So now the cell will become the haploid, because in meiosis first is a reduction division, means it decreases the number of chromosome by the half amount. So now will be one chromosome in each cell. Suppose this is A cell and B cell. So A cell have one chromosome, B cell have one chromosome. So finally two cells are formed which are haploid. Now we will start the meiosis second. We completed the meiosis first here, all the steps. Now the meiosis second. So meiosis second is the equational division. There is no again reduction in chromosomes, and this occurs into the haploid cell, because now these cells are haploid. So meiosis second is occurring in the haploid cell, not in the diploid cell. There is a short step, that is interkinesis. Interkinesis means a very short resting period between meiosis first and meiosis second, that is the interkinesis. Now the meiosis second have karyokinesis and cytokinesis second. Karyokinesis second again have 4 stages, that is prophase second, metaphase second, anaphase second and telophase second. Now these all the stages are same as the mitosis or like of mitosis. So these are very simple steps. You have actually seen the mitosis, so that's why you can easily understand the prophase second, metaphase second, anaphase second and telophase second. And then finally the cytokinesis second. Then prophase first, we will start with the prophase second. So in prophase second there is a nucleolus and nuclear membrane disappear. Now you have seen in mitosis, in prophase there is a disappearance of nucleolus and nuclear membrane. Same things are happening. Chromosomes become compact, means short and thick. Same process will be happening. Then centrioles start to migrate towards the opposite pole, which is happening into the prophase of the mitosis. So one chromosome, A cell and B cell. A cell have blue chromosome. For our understanding, we are remembering the color. A cell have blue chromosome, B cell have black chromosome, and both these chromosomes are with the exchange chromatids, exchange material. Now prophase, after the prophase there is metaphase second. Metaphase, the characteristic of metaphase is the alignation of the chromosome at the equatorial plane. So simply what happened, these chromosomes, now we are showing only the one chromosome because we have taken the 2 chromosome as a diploid condition. But with respect to human beings, in human being there is a diploid condition, that is the 46 chromosome, and it means that in haploid condition there will be a 23 chromosomes. So all the 23 chromosomes will align at the equatorial plane. Suppose 23 here and 23 here, but we are considering the one chromosome as a haploid condition for our understanding, because we cannot represent all the 23 chromosomes because it will become very complicated. So chromosomes at the equatorial plane, key point, that is chromosomes will align at the equatorial plane and spindle fibers will get attached to the centromeres. So this is the end of metaphase second. Now the next, that is anaphase second. What happened in the anaphase second? In anaphase second, now there is a splitting of the centromere, because in the mitosis, in anaphase there is a splitting of centromere. So all these 4 stages, that is prophase second, metaphase second, anaphase second and telophase second, are same with the mitosis. But prophase 1, metaphase 1, anaphase 1 and telophase 1 are not same with the mitosis. So splitting of centromere is occurring in the anaphase second, but splitting of centromere does not occurring into the anaphase first. You have to remember this point. So due to the splitting of the centromere, what will happen? Now the two chromatids, each centromere have two chromatids, but now in anaphase 1, each one centromere have one chromatid, because centromere is now splitted, and due to the splitting of centromere, each centromere have only one chromatid with the exchange material. So each chromatid moves towards the opposite pole into the anaphase. Second, the forces, you know that the forces, the chromosomal fiber and the interchromosomal fiber. Chromosomal fiber contracts and interchromosomal fiber stretches. And the last phase, that is the telophase second. So what happen actually into the telophase second? I am showing the completely, that is telophase second as well as the cytokinesis second. In the telophase, what will happen? These chromosomes or chromatids completely reach to the respective poles, and nuclear membrane will start to appear, nucleolus start to appear, and finally in then in cytokinesis second, what will happen? The nuclear membrane is completely appear, nucleolus is completely appear, and the cytoplasm is divided, and the two separate cells will be formed in the cytokinesis second. So I am showing completely, that is telophase second as well as the cytokinesis second. So A cell, we are considering in A cell, in A cell there is a blue chromosome. Here I am not showing the B cell. In this diagram also, in anaphase and metaphase, I am not showing the B cell, because these processes which are happening into the A cell, which are also happening in the B cell. So there is not necessary to represent here, because you can understand from this A cell. So in the in A cell, chromosomes are aligned at the equatorial plane. Same in the B cell, chromosome will align at the equatorial plane. Then same splitting of centromere and movement will be happen into the B cell, and same separation will happen, cytokinesis will happen. So finally A1 and A2 means A cell produces again a two cell from one cell with a one chromosome. Now these single chromatids will separately act as a one chromosome. So this is one chromosome with one chromatid, this is one chromosome with one chromatid. So now this is also haploid N, means chromosome 1. So no reduction in number of chromosome. Here is 1. Same in the B cell also, B1 cell and B2 cell will produce. So one chromosome. So finally there is a formation of 4 cells, that is 1, 2, 3 and 4. So we start with a one cell which have 2n is equal to 2c, and from these we get the 4 cells with n is equal to 1c, 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 one chromosome. Understand? One chromosome, and now these are haploid, because parent cell have 2c but the daughter cell have 1c, that is why haploid. But these daughter cells which are not exactly same, because they have the exchange material, but in mitosis the daughter cells are exactly similar with each other, and they are exactly copy of their parents, because there is no exchange of material. But in meiosis, these daughter cells are not exactly similar with the parent cell. They have a variation, they have a different characteristics because of the mixing of the genetic material, exchange of material into the prophase first due to the liptoteny, zygoteny, pachyteny, diploteny and dikinesis stage. Due to the crossing over, there is exchange of genetic material, and now each cell have the exchanged chromatids, and that's why each cell have different characteristics, different genes, different combination of the genes. That's why this cell have this meiosis produces the variation. So here we completed the meiosis process, complete meiosis process. Last topic is that the significance. So the significance of meiosis is meiosis produces the haploid gametes, that is N. From 2n it produces the haploid gametes during the sexual reproduction. that is the haploid condition in human being, that is the 23. Means the sperm of the male of the human contains the 23 chromosome, and the ovum, that is the gamete of female, contains the 23 chromosome, and when the sperm and ovum fuses, it will produces again an organism with the 46 chromosome. Due to mixing 23 plus 23, it will become 46. So the embryo or the fetus will have 46 chromosome. So due to meiosis, the organisms can maintain their chromosome number. If there is no meiosis, then the chromosome number will get double. If there is an absence of meiosis, what will happen? If the cell divides or gametes are produced by the mitosis, what will happen? The gametes have the 46 chromosome, because chromosome number will not reduce, because there is no meiosis. Mitosis is a equational division. So 46 in the male gamete, 46 into the female gamete. When there is a fusion, it will become 92 chromosome. So it will change the species number, and as the chromosome number of the species changes, it changes the characteristics of the species. So all the characteristics of the human being, and there will be a deformity into that human beings. So that's why meiosis is very important to maintain the chromosome number of the species. So that's why due to this meiosis process, all the human beings have maintained their chromosome number, that is 46. So each and every individual have the 46 chromosome. So this is the main significance of the meiosis process. So here students, we are completed our chapter, that is cell cycle and cell division. In the next lecture, we will start the new chapter. Thank you.